Okay, thank you very much, Gary. Thank you very much, y'all, for coming this evening. I've been looking forward to this, and I'm very excited to have this opportunity to talk to you about some of the things that I've been doing in my research program and the people that have been working with me over these years, and also the chance to talk about the Year of the Sea and the capabilities that we have for marine science here at Heriot-Watt. I didn't anticipate that introduction to be that long. It was simply some information that I provided for Gary to use, but apparently he just went ahead and read the whole thing. This is not Nemo. This is a zebrafish, and I would be remiss if I didn't start out this lecture with an acknowledgement of this fish, because it's a very important fish. It is it's become a model of research for both biomedical research and environmental science. We have a zebrafish facility here at Heriot-Watt University, which is the primary focus of my laboratory. It's an amazing animal. About 1 to 2 inches in total length is the length of the adult. You notice here that the embryos, they have a clear shell. You can see right through the shell, and from fertilization to hatching 72 hours later, you can watch the whole developmental process. It's an amazing scenario, and it presents a unique aspect for study. There's been a considerable amount of research done with this organism. The genome has been sequenced, and so there's lots of tools that we can apply to conduct studies with it. Now, have a look at this. So some of you who are familiar with balancing childcare responsibilities with academic responsibilities recognize that at times our offices become childcare centers. And in this case, I went to a meeting, and I was slow coming back from my meeting, and my daughter was in the office a bit grumbly because apparently it took longer than I had anticipated to get back. And I looked at the whiteboard, and she had drawn this cartoon on the whiteboard. And it's interesting when you think about your children and how they have a perception of what you do, and that might be quite different than what you think you do yourself. But in any case, I was looking at this, and I'm going to talk about overinterpretation of data later in this talk, but we're not going to take that in this context because I'm the father and I am going to overinterpret it. But look at this. So here is apparently my hand dumping some toxicants in a tank of water where there are some zebrafish. Okay, and I added some things to this. So this was her writing, but I put it in type so that you could read it in this slide. But the interesting thing that I wanted to point out when I was looking at this was this difference between local and global perspectives. And so we have some fish here right next to the output of this toxic substance into the water. This one is very unhappy. This one's starting to detect the sense of this toxicant. This guy's getting out of the area. These guys are over here. They're laughing at their comrades who are being subject to this. But I found this one particularly interesting. This one is actually sitting on a stool with some popcorn, watching it as if it were a movie, separate from the event that's occurring. And I think that's important in the context of environmental toxicology that we recognize that we're all within the world, and things that are done at a local level affect all of us. So if there is an environmental toxicology issue in some remote region of the world, those types of things can have knock-on effects that affect us. I think that's what you're intending, right? Okay, so now when we think about some of the environmental successes that we have, this is an image that came out in the Daily Mail, which was pointing out that an environmental success here in the UK, a heavily polluted river that has been converted into improved to the extent that now we have salmon running in that river, and it's been recognized as one of the best for salmon fishing in the UK. So that's very, very good. We're very proud of that. However, are these local successes in environmental protection on the back of a global failure? Is it because we're simply exporting our environmental problems, waste disposal to other countries, locating our hazardous industries in other countries, allowing the use of banned substances, producing banned substances? and allowing their use to occur in other substances. We need to care about this. These are things that can come back and have an effect on our global environment. Okay, so in 2009, Rockstrom and co-authors published an article in Nature where they identified this concept of planetary boundaries, and they identified 9 environmental priorities that are indicated here. Things, the big ones: climate change, ocean acidification, ozone depletion, the nitrogen cycle, freshwater use, land use, biodiversity, atmospheric loading, and over here is chemical pollution. And the concept that they were getting at was that they were trying to recognize that there's some critical values at which, in the context of the biophysical processes of the planet, that once those are passed, the critical value is passed, that there will be irreversible change in that planetary system. So if we take, for example, climate change, and we recognize, okay, we've all heard about carbon dioxide releases, and a critical value of carbon dioxide concentration in the atmosphere. Once that critical value is passed, we can anticipate that there will be irreversible changes in climate with considerable negative effects potentially on humanity. And so this article was recognizing these 9 priorities. In the case of chemical pollution, one of these 9 priorities, it's not quantified yet. So what is the critical value for the amount of toxicants that can be released into the environment before we will see irreversible changes that will have impacts on the global humanity? We can recognize individual environments, local areas where the contaminant loads have compromised particular situations, but I'm talking about planetary boundaries where the functioning of particular systems within the planet will be affected. They identify some of the big toxicants of interest: persistent organic toxicants, plastics. I'm going to talk about plastics a fair amount today. Endocrine disruptors, heavy metals, nuclear waste. Okay, so how do we address these? We know there's toxic chemicals. There's new chemicals produced every day, numerous each year, and they have some toxicity associated with them that needs to be assessed. If we consider our concentration response profile, if we recognize as far back as Paracelsus in the 1500s recognized that everything is toxic, it's just a question of dose. So if it's a low, a substance that is rather benign, eventually the concentration is high enough, it will become toxic. So this is a rather simple scenario. We increase the concentration, the response increases until we get 100% response. Rapidly gets more complicated than that, because then we have some questions. Well, which response is relevant? So is it something as crude as mortality? Does this substance kill the organism? Or more subtle effects? Is that what we should be looking at? Which organism is relevant? How long should the exposure duration be? What concentration is relevant? So we have concentrations in the environment. They may be very low. How do we understand what the effects of those concentrations are? Okay, so unexpected toxicity. This situation comes in, how do we recognize whether we're investigating the right effect, the right response? And this was one of my early projects. It was a NOAA EcoHab project, and we were investigating the toxicology of the cyanobacteria microcystis. This is a cyanobacteria that produces these blooms on the surface of the water. So when you see fresh water with this green appearance, quite likely there's some microcystis there. It occurs when in eutrophic environments where there's a lot of nutrients in the water. We know that this cyanobacteria can produce toxins that are liver toxins called microcystins. But the cyanobacteria produces a lot of other substances, and we were interested in finding out what other effects those substances might have. So how do we engage in that? So first thing, well, we have our zebrafish model. So these are larval zebrafish. This guy's about 72 hours from fertilization. And put them in these different exposure scenarios with the cyanobacteria, extract the RNA out, and then investigate the global gene expression. So the global gene expression is an approach that allows us to interrogate the expression of all of the genes in the organism at the same time. It's very useful when you don't know what is going to happen. You can identify which of those genes are of interest. So we evaluate which genes are switched on. And one of the ones that we found that we were especially interested in was this vitellogenin gene. And this is a gene that codes for a yolk protein. So in female fish, when the endogenous estrogen, which is the same 17-beta estradiol that we have in humans, when that's released, that female fish, the estrogen binds to the estrogen receptor and induces the transcription of vitellogenin, leading to the production of yolk, which is then deposited in the eggs of the developing fish. But we find that these genes are turned on in these larval fish, which is an indication of an estrogenic response. So it was the first report of estrogenic activity by a cyanobacteria. We didn't anticipate this. This occurred because we did this global gene expression approach. Had very interesting implications because it said, okay, well, in the environment, there's natural endocrine disruptors. And perhaps some of the reports of feminization of fish in the natural environment could be because of natural substances from cyanobacteria rather than what we typically think of, which is release of anthropogenic substances that have estrogenic activity. Okay, which organism? So we have to make a decision about which organism we're interested in. I've already introduced the zebrafish model. Zebrafish is very good for some things, not good for other things. It doesn't live very well in Scotland. It needs to have temperature up above 20 degrees, otherwise it does very poorly. We use this model to essentially identify some interesting pathways of toxicology, and then in some cases we'll apply it to other species of interest. This is Lombardi, which is a fish in Brazil that we've been working with, trying to develop native species that are representative of the environment in Brazil. Along those lines, we've also been working with an amphipod in Brazil with my close friend and colleague, Professor Gisela Umbizito, and her students, Mariana Monizi and Amanda. And those students have come to my lab here and worked also on this organism. And fortunately, I'm a fish toxicologist. Fortunately, we have Liz Dorinda, who's in the audience, that is an invertebrate immunologist who actually knows about these organisms. I can do their genes and a few other things, but otherwise, I don't know too much about them. And I've even started to work on these sponges, which is absolutely amazing to me as a fish biologist that there's some sponges. They have RNA in them, and apparently they have genes as well. We're identifying gene expression pathways that occur from these sponges. And so that has been a fascinating development that Joanne Vad, who's one of my PhD students, is working on in the context of oil pollution. And her supervisor, Murray Roberts, is also in the audience today. Okay, now, concentration. And so we've talked about which response to address, which organism to address, and the concentration is another issue. So how do we define which concentrations are relevant? A lot of the laboratory toxicology studies are using concentrations that are well above environmentally relevant. We can identify toxicity, but it doesn't help us inform what's actually happening in the environment in many cases. Particles, I'm going to talk a lot about plastic particles today and nanoparticles. And particles present a unique challenge for toxicology. And I'll remind you, here's our dose-response relationship. Concentration increases. We'd love to have this beautiful, smooth, sigmoidal-shaped curve that shows the effect. But when we do copper nanoparticles, we know they're toxic. But look at these 3 separate experiments. This is not a nice concentration response. Simple mortality of an endpoint for these zebrafish. What we notice is it's not reproducible, kind of goes up, kind of goes down. Very troublesome. The reason is that the nanoparticles don't stay in the aqueous phase in a homogeneous way, like a traditional dissolved substance. So the exposure is not consistent from one concentration to another. And so that adds a complexity when we're trying to understand the toxicology of these particles. Now, we're going to talk about particles, so let's start with a bit of information about particle sizes. Okay, so here's some plastic microspheres. Some of these microspheres are used in products, facial scrubs and other types of things. 1 millimeter in size. So you can see a size of a millimeter. This is in a human hand holding these particles. And then we have a neutrophil, which is a blood cell. It's about 10 microns in size. And when we think of a nanoparticle, a nanometer, this is a C60 fullerene, which I'm going to talk about in a minute. This is about 1 nanometer in size. And when we think about the relative size of this, the relative size of this fullerene relative to the football is the same as the relative size of the football to the entire planet Earth. So that gives you an idea of how small that nanometer is. Okay, so shortly after I took my first academic position at the University of Tennessee as a toxicologist, this area of nanotoxicology was developing quite swiftly. So there was nanotechnology, the design of engineered nanomaterials that had unique properties. And there was concern emerging about the potential that these nanomaterials with their unique properties would have unique effects toxicologically if they were accidentally released into the environment. There was a lot of speculation about this, a lot of concern, but very little evidence at this point in time. Some of these were pointed out in some of the prominent articles at the time. And while there was recognition of there's a lot of potential benefits for humanity of nanotechnology, that we can do things with less material, and we can have lower amounts of waste and take advantage of the unique properties that these materials have. Are the risks, do they outweigh those benefits? There was calls on specific regulation. Could nanotechnology go down the road of genetically modified organisms wherein we might not get the full benefits of the technology? Other articles came out to point out, well, in the toxicology sense, we need to be considering a list of properties of the particles themselves that we might be able to relate to the toxicity. Things like the size, surface area, functionalization, surface charge, porosity. We were led to believe that there would be nanospecific effects, and that those nanospecific effects are things that we should be investigating and identifying within our investigations. There was numerous review articles that speculated about the potential toxicity. It seemed like every journal was having a special issue on nanotoxicology where there would be all of these review articles written, and nobody was actually doing any experimentation. So there was a lot of speculation and very little evidence. So I got involved in the situation investigating the C60 fullerene. It's a beautiful molecule. Look at that. I mean, even if you don't like football, it's a beautiful molecule, right? Look at that. 60 carbon atoms that are arranged in this spherical cage-like structure. It was first manufactured in the 80s, Nobel Prize in Chemistry, has very unique properties, a little less than a nanometer in diameter, has these partially delocalized pi electrons, which give it some unique properties, can absorb light, produce reactive oxygen species under certain conditions. Generation of reactive oxygen species raises a concern in the toxicological sense because reactive oxygen species can react with biological molecules and cause toxicity. So, speculation that it might be toxic, C60, perception then is, oh, it must be toxic. And evidence, you kind of don't recognize that. Okay, maybe there must be some evidence. Everybody's talking about it, right? And then there was this report in 2004 that C60 caused toxicity in the brain of bats. And naturally, when there's a new material that has projected use, and here we have a situation where a report is saying brain toxicity is one outcome of exposure to this substance, raises a lot of concern, raises a lot of interest. Now, it led to the speculation that the C60 was absorbed by the organism, was transported to the brain, right? So this was an aqueous exposure, absorbed across the epithelial membrane into the blood, transported to the brain, generated reactive oxygen species, and that caused oxidation of biomolecules in the brain that led to this detection of toxicity. Okay, but As I mentioned when I started talking about these engineered nanomaterials, they're problematic to study. And C60 itself is extremely insoluble in water. So you have to do some tricks to get it to go into water to facilitate the aqueous exposure. One of them is to first dissolve it in an organic solvent. So take your C60, which is a black powder, put it in the tetrahydrofuran vehicle solvent, it dissolves, and then take that mixture of C60 and THF and put it in water, and then use a rotovap system to evaporate away the THF, and then you're left with aqueous agglomerates of C60. So they're a couple hundred nanometers in diameter, consisting of many C60 molecules. And this is what the exposure that was done in this study. So we started investigating it in this situation. We used our zebrafish. We exposed them to different treatments. C60, where we just put it in the water and sonicated, and it stirred it over a long period of time. And this also allowed the formation of these agglomerates of C60, although they were a little more heterogeneous in terms of their size. We had our THF C60 treatment and a THF vehicle control and a vehicle control. We started noticing that there were effects in our THF vehicle control. And when we looked at the previous study, they hadn't included a THF vehicle control. We exposed our zebrafish, did our global gene expression analysis, and when we looked at the pattern of gene expression between the THF C60 and the THF vehicle control treatment, they were essentially identical, which said that the C60 itself was not contributing any toxicity in that situation. This included genes that were responsible for responding to oxidative stress. These results showed that the C60 was not responsible for this oxidative injury that was in the brain. And this also created some controversy within the nanotoxicology community. And a number of other studies went on to test the same thing and demonstrate that this was in fact the case, that the THF vehicle and the THF C60 was leading to the production of decomposition products that caused this effect. It wasn't because of the C60. Now, just this week, I was curious to see how the progression of this information in the literature. So I looked at the Web of Science citations. And this is a plot of that first article and our article. Now, I'll say that these articles were both published in the journal Environmental Health Perspectives, which is a prominent journal that's published by the National Institutes of Environmental Health in the US. And look at this, the number of citations that this article, which documented the presence of toxicity in the brain of these bass. Even after our article came out, which showed, well, it's actually the vehicle, and it was corroborated by numerous other independent studies. Still, in 2017, that article is getting more citations than the one that's showing there's no toxicity. Why is that? It's because people were more excited about toxicity than no toxicity, right? And there's another few examples, and I'm going to talk about that. And this is one of the main points that I'm going to make in this presentation, is that we have to be careful of that particular aspect. Now, looking at carbon nanotubes, same situation to a less dramatic extent. There were reports of carbon nanotubes, which is essentially a C60 fullerene that's elongated with the ends open. And there was toxicity reported. Abnormalities, again, in the brain. These consisted of vascular changes in the brain that were detected on histology. Some changes in fish behavior. But when we started doing experiments with this, we noticed that, gosh, look at the gills of these fish. They're coated with all of this black material. To get the carbon tubes into the water, they had to be coated with essentially a surfactant, which is like a soap, to make sure these agglomerates would stay in the water, because the tubes themselves are extremely insoluble in water. Okay, so when you see this occlusion of gills, you'd expect there to be some kind of response in the fish that's a consequence of that. It's a much different situation than a nanospecific effect with a nanomaterial that's getting absorbed, transported to a tissue, and causing a toxic effect by a specific mechanism of action, which is what we might expect from another type of toxicant. Okay, so we investigated this further and found even with a diet, so we looked at dietary exposure, high concentration, 500 milligrams per kilogram. In that situation, the food is black. We don't get any toxicity. No abnormalities in the brain. My background is in fish histopathology. I focused on the brain. I was looking at those tissues. I wanted to make sure. Didn't see anything. No changes in fish behavior. We repeated the aqueous exposure and documented this respiratory distress, looked at the gills. Yes, in fact, this type of aqueous exposure causes lesions in the gills, which are consequence of this accumulation of material on the surface of the gills. This work was done by Dr. David Boyle, who was a postdoc with me down at University of Plymouth. He's just recently joined my group here at Harriet Watt, so I'm really pleased to have him here and working on the group on these particle toxicology questions. Okay, so for all of the investigations that we have done with a variety of different nanoparticles, we have not found in aquatic organisms anything that supports a nanospecific effect in terms of toxicity. That's not to say we haven't found some interesting things along the way. One of the things is this concept of sorption and desorption. So remember, these are particles in the water. So you have this particle in the water, and things will stick to it, and things will come off of it. And so I like to present this as an equilibrium expression, where you have the particle and the toxicant in the aqueous phase. This is in equilibrium to the particle with sorbed toxicant to its surface. So as you might expect, the particle has a surface charge. It has surface properties, and that will affect what types of substances will sorb to it and partition onto it. Sorption, it is a critical aspect of toxicology. Okay, it can impact how toxicants are transported around the environment. It can influence organism exposure if those substances are transported around the environment because they're riding on particles. There's lots of natural particles in the environment. Sorption is a very important process in that sense. So our role in this was to use our zebrafish model and link a gene expression response to this bioavailable fraction of the toxicants so that we could interrogate within this aqueous phase the amount of substance that was available and not sorbed, and manipulate the conditions to detect changes in the sorption-desorption processes. This provided some unique insight onto this. Okay, now, I'm not going to go into all the details, but we've had some really interesting projects on this. Danae Patsu is a PhD student who's just finished with me, and she's worked a lot on various nanoparticles, the sorption processes, started to apply ultraviolet lights in this scenario to see how these interactions between sorption, desorption, and this complexity. One day, I was in my office early. There was a fire alarm. I went outside, and I happened to be standing next to Martin McCustra at the area where there was the fire alarm, where we had to sort of assemble. And I was chatting with him, and he was interested in particles, and I was saying I was interested in particles. He's here at Harriet Watt University as an astrochemist, astrophysicist, well out of my league in terms of his understanding of all of this stuff. And anyway, from that conversation, we went and had a coffee, and we wrote this project into the Europath project. We got that funded, and now we have Lindsay St. Mary, who's a new PhD student who's taking this work forward, looking at the partitioning of polycyclic aromatic hydrocarbons onto nanoparticles that are representative of interstellar dust, and looking at the sorption-desorption phenomena. Particles, as I indicated, are very important. Also, very present in indoor dust. And so we've taken these techniques with our zebrafish and bioavailability model, and with my colleague, Dr. Miranda Lowe at the Institute of Occupational Medicine, we're investigating the bioactivity of substances that are associated with indoor dust samples. Okay, to shift gears into plastics now. Look at this, plastics. A lot of attention on plastics. I mean, probably all of you have seen recent news reports about plastics in the environment. And when we see pictures like this, they're very dramatic, very evocative pictures, right? A seal with some net around him, turtle that's obviously grown with a six-pack casing attached to it. Very dramatic, right? Okay, so a lot of attention on plastics right now. There's a lot of plastics that are produced. They're produced as small pieces of plastic. They're used in products that we are using every day. Hundreds of millions of tons produced every day, 7,000 million tons produced since 1950, a lot of it. Most of this is a waste management issue. This is where our local policies of waste management have global implications. Microplastics. These are small pieces of plastic. Some of them are manufactured as microplastic particles for use in products as such. Other times they fragment from larger pieces of plastics to form these small microplastic types. A lot of different shapes of the primary polymers that are used to make the primary plastics. Okay, where do they go in the ocean? So when they're released into the ocean, there's large accumulations found in some of the gyres, North Pacific gyre in particular, but in other places where the currents cause this circulation and concentration of the materials in these locations. Okay, so now, within that context, there's been plenty of speculation about the effects of plastic. We don't have any idea about how much plastic we can release into the environment before we reach this critical point of a planetary boundary, which I indicated in one of the first slides. There's some questions that we might want to address and think about the evidence. Are microplastics ingested? That's one question we might care about. Are they absorbed and accumulate internally? So when I say absorbed, I mean not just present within the gut, but actually ingested, absorbed across the wall of the gut, and then internalized into internal tissues. If they're transferred in the food web, is there a risk of human exposure? Should we care about that? Are there substances attached to the microplastics? And are these substances potentially toxicants, bioavailable to organisms? We need to recognize that there is evidence on these questions already. Okay, despite plenty of speculation that's coming out, we need to recognize that we do have some evidence to recognize. Okay, I'm going to talk about some studies that we have done and refer to some others in the literature as well. Ingestion of microplastics. Clearly, organisms ingest microplastics. This is very widely recognized. No question about that. If it's in a food particle, they will ingest it. This is one of my favorite studies. This was done by an undergraduate student that worked with me, Joseph Hatfield. And we were trying to address, okay, will they ingest the microplastics, and what happens when they ingest them? Will they pass through the gut, or will they be retained? And so, how long does it take? So our approach was to prepare some feed pellets. These were feed pellets, or fish paste, and we put in the microplastics. And we had treatments with microplastics of different sizes and equivalent pieces of sand that were the same size as the microplastics. And he counted the plastics particles, each particle that he put in the pellet. So there's no question about how many particles are in there. Fed the pellet to the crab, and then counted the pellets, the plastic particles that were released into the feces over time. And all of them that came out. Very tedious process, and he did it. Marvelous. Okay? So, what did he find was that all of the microplastics were found in the feces within 3 days. That's all of them. So if you wonder, well, maybe some of them were absorbed, all of them came out. We counted them in the beginning. They came out. The microplastics stayed in the gut a few hours longer than the sand particles. So you're sitting there thinking about a crab like this. I mean, how many particles does that crab confront on a daily basis every time the tide changed? There's a lot of particles there. Okay? So is there something special about microplastics that means that they're going to, you'd expect the crab to be pretty good at avoiding absorption of small particles. And we found it did in that case. This is another interesting story, one of my favorite studies. This is Dr. Anna Katerina, who's a postdoc in my group. She joined probably 4 years ago as a Marie Curie Fellow and has stayed as a named postdoc on the NERC project. And she's been going around Scotland measuring the amount of microplastics that are in wild mussels. And So, okay, why would we do that? Well, mussels, of course, filter a large amount of water. So if there's microplastics in the water and you have a filter-feeding organism, we could presume that that type of organism is going to accumulate large numbers of plastics. We eat mussels, okay? So what she did was collect these mussels, digest them, count the microplastics that were present in them, and we were getting calls from various journalists who were wanting us to make comments about the risk of human exposure from mussels. And I got a bit frustrated about that, and I said, well, let's attack this in with a little bit of evidence. And she came up with this idea to investigate how many microplastics are there just in the house dust. So say you're preparing a plate of mussels, and what Anna did was to evaluate how many microplastic particles fall from the dust just within your house during that preparation and also next to your plate when you're eating. And she sent us these things to sample. We took them home and sampled the dust during this process. We found that there was an average of 3 microplastic particles per gram of mussel, and about 100 or so we would be exposed to during that meal. And so there's many, many more microplastic fibers that you're exposed to just from the dust falling in your house, okay? And so when the journalists ask me, and I say that, they never quote it. You know, it surprises me. I'm like, well, here's the information. I mean, how worried are you about the dust particles that you ingest in your house? But mussels, you know, better think about that, right? Okay, so toxicant sorption. This is something about plastics that we care about. Plastics, they sorb different substances. Within the environment, people have gone out and collected plastics and removed toxicants from them and analyzed them, found a large variety of different substances attached to plastics. But the reality is they're very low concentrations, and we have to remember that just because they're present does not mean that they're having any toxicological effect. Some studies have shown that actually, in one interesting study that went out and sampled seabirds and found that seabirds that had larger amounts of plastic in their gut had lower levels of persistent organic substances in their tissues. And they offered as an explanation that the plastics were actually sorbing the toxic substance as it passed through the gut and released from the bird. Our results indicate, yeah, microplastics sorb a variety of substances that we've challenged them with. We haven't been able to see any delivery of these toxicants to organisms. So one way to look at it from our perspective, when plastics in the environment actually removes the toxicants, holds them, makes them less bioavailable. Presence of plastics, you could argue, reduces toxicant bioavailability. Okay, so we're working on this nanotoxicology, nanoplastics, so microplastics and nanoplastics down into the size of 100 nanometer size plastics. This is one gap in information. I will say that small pieces of plastic, we're still trying to recognize whether those particles can be absorbed or not. Can they get across biological membranes? Can they accumulate? We have an experiment that's going to occur in a couple of weeks with our collaborators from Plymouth, Richard Thompson and Steve Rowland. The postdoc there, Maya, has been able to make C14 labeled polystyrene nanoplastics. And we're going to do these exposures with fish and document whether these particles can be absorbed and accumulate in tissues. Another thing that we've worked on here with Tony Gutierrez, which is here at Heriot-Watt, he's a microbial ecologist. His postdoc on the project, Dr. Steve Summers, has been investigating what I call microorganglomerates of plastic particles. So when we think about plastic particles, we often think about a pristine plastic particle, clean thing. What we recognized is that actually in the environment there's a lot of things out there. There's a lot of substances, there's a lot of microbes, And if we think about our microorganomerate, depending on the size of the plastic may be much smaller than a microbe, or it might be much bigger, and the microbes might be attached to the surface. And so these agglomerates are going to be some kind of a very complex assortment of substances and organisms, and that's what's actually present out there in the environment. Steve has done this. He's shown that these agglomerates form in natural seawater, so these small particles agglomerate according to the substances that are produced by the microorganisms. This is what they're actually looking like by scanning electron microscopy. There's a lot of stuff on them, okay? So that affects their behavior. That affects their physical chemical properties, their buoyancy, and how they're going to be transported around in the environment. So recently, in fact, just last week, we put in a proposal that's based on these thoughts and where we think there are existing gaps in understanding of the effects of microplastics in the environment. It's called Tap to Trench. And this is starting with a focus on microplastics that are in wastewaters, how they're transported into the environment. We're using the River Clyde as our model system, and then modeling the transport of those into the marine environment, and then ultimately to the deep sea environment. We proposed a NERC ship to go out and sample these areas. So our belief is that the deep sea environment is an area where microplastics are likely to accumulate to high levels. Over long periods of time, that level can increase and increase and increase. And at some point, we hypothesize that there might be significant effects on ecosystem processes, things that occur in the sediments, how sediments process nutrients and large-scale types of things, and also critical habitat-forming organisms like deep-sea sponges and corals. So this is the type of project that I'm very excited about here at Heriot-Watt University, because it takes advantage of some of the key features that we have. So within the investigator team, we have people from different schools. We have some of our new Lyle appointments that we're very pleased about. We have Murray Roberts and Sebastian Hennig from Edinburgh University, bringing expertise on corals and sponges. And we're also taking advantage of the Wolf Summit Aquarium, which is a phenomenal aquarium facility that is right here on campus. At the same time, we have the St. Abbs Marine Station, which provides a very important link between the laboratory and the real marine environment. Another project, I do a lot of work in Brazil. Just a quick mention that we're very interested in how microplastics are transported within riverine environments. And this is looking at some of the prominent rivers in Brazil with my colleague, Dr. Cassiano Raimundo, who is at the Institute of Chemistry at the University of Campinas in Sao Paulo State. This is a picture of the meeting of the waters of the Amazon River. Beautiful spot from an aquatic science perspective. You've got the Rio Negro, which has a low pH coming into the Rio Solimões, however you say it properly. And you have this meeting of the waters, which occurs with the different physical chemistry, different temperature, and different water chemistry characteristics. Now, here we are at Heriot-Watt University. It is the year of the sea, and we're very excited about this as marine biologists, marine scientists. And we have some very unique features that we can use and that have developed even over the 5 years that I have been here at Heriot-Watt. One of them that it's phenomenal. If you haven't been to St. Abbs before, go there. It's a beautiful place. First time I went there was for a short meeting. A few weeks later, I took my family there because it's such a beautiful location on the coast of Scotland. We have co-directors, Dr. Karen Deal, who's at Napier, and Dr. Bill Sanderson, who's here at Heriot-Watt University. This is a family charity set up by the Nesbitt family. There's the St. Abbs Marine Station, and also very importantly, there is the St. Abbs Visitor Center. This provides a unique ability to engage directly with public outreach. So in our NERC proposal, we make use both of the Marine Station and the St. Abbs facility. Some of the other things that we have at Heriot-Watt, the Isle staff, the existing staff, some of our new appointments within the Lyle Center have really expanded the capability that we can apply to address real problems within the marine environment. We have unique facilities in the Wolfson and John Muir Aquarium, Lyle Center equipment, the Orkney campus with its ability to engage in blue economy aspects. We're starting to engage in Malaysia and Dubai. I've got work to do there, but we believe that there's, in the future, some potential to develop marine science angles there. It's not just about research. The interest in an international marine science program has gotten off the ground. We have a vision of taking advantage of our Harriet Watt International University of the Year and capitalizing on this in the context of marine science. And perhaps in the future we can have marine science degrees where students are able to move around institutions and have experiences in marine science in various locations. So, challenges and opportunities. Marine science is clearly of global importance, and I believe that Harriet Watt is poised to contribute to these key global environmental challenges. And I am very excited about that. I'm very excited to be here. Last point on my particular presentation, remember to consider the evidence, okay? Students who are looking at the literature, look at all of it. Try to understand, okay, it's not just about picking that one that supports a particular angle. Watch out for speculation. Recognize when you are speculating, recognize what is supported by evidence. And then, just because we're approaching the question and answer session, I put this last point, which is one of my favorites from Barack Obama, which says, be kind and be useful. Okay, in the acknowledgements, I've acknowledged, we've been fortunate to have a lot of funding over the years from different sources. And so those are there. I acknowledge individuals who have contributed to the research program over the years. But a special acknowledgement I want to make is to my family, some of whom are here, and my extended family back in the United States. But I never really recognized some of the other ones. Scott Cooper was my supervisor when I was an undergraduate student. He was influential to me. Elise Irwin was my supervisor as a master's student. John Grizzle, my PhD supervisor. Marcia Black was my supervisor as a postdoc. And then Dr. Gary Saylor at the Center for Environmental Biotechnology and the Forestry, Wildlife, and Fisheries, Richard Strange. These are individuals that were key to my development, and I wouldn't be here without them. Thank you very much.